So some information about me. I'm Shirley Liu. I'm a professor of biostatistics and computational biology at the Department of Data Science at Dana-Farber Cancer Institute. I'm also a professor at the Harvard School of Public Health and also in statistics department at Harvard University. That's why this course has a STAT115 title. So my research is on computational biology in translational cancer research. We work on gene regulation, transcription epigenetic gene regulation at the bulk level or single cell level. We also use CRISPR screens to look at gene regulation. And recently we are heavily invested in understanding cancer immunology. So if you want to know more information, you can go to the lab website. We'll also post a video about our introduction. And so this year, because of COVID, both FAS and Harvard School of Public Health were able to allocate more TAs to make sure that students who are taking the course remote will have enough help. So as a result, we have 6 TAs sign up for this course, and they will be hosting the labs for every week and also on the weekend office hours in order to answer questions you have regarding the labs and also help you with homework. Okay, so I'm sure you will be working a lot with them. There are some information on the course organization. This year, we're going to divide the course into 4 different modules. The first module is on RNA sequence analysis and sample classification. You will learn read mapping, how to do differential gene expression, how to cluster the samples, look at the gene annotation of a group of genes, or and also remove batch effect. Sorry, there's a typo here. And also how to do dimension reduction to cluster the samples, and also learn supervised machine learning to classify samples, for example, to see whether a new sample is normal or diseased for diagnosis. In module 2, we will cover transcriptional and epigenetic gene regulation. So, basically module 1 will show you these genes are differentially expressed, but then the second module will tell you why they are differentially expressed, right? So here we cover transcription factor motif finding, ChIP-seq, DNA methylation, histone modification, how to use hidden Markov models to annotate genome segments, and chromatin accessibility and higher order chromatin interaction, and how to use these type of data to understand gene regulation. The third module, we will cover GWAS interpretation and single cell data analysis. There are courses, a whole class, a semester course to cover GWAS. So we are only going to use 2 lectures to cover this. So we'll make them really brief to kind of, basic information to give you a basic idea of what is a GWAS study, what is SNPs, LDs, what are some of the public resources to evaluate GWAS, and how people use functional genomics data or epigenetics data to interpret a GWAS result. And for single cell analysis this year, we are going to increase this to 3 lectures, 2 on single cell RNA-seq analysis and 1 on single cell attack-seq for understanding gene regulation. And the last module is going to be on cancer genomics and cancer immunology. We have a pretty heavy coverage on cancer, partly because I work at Dana-Farber Cancer Institute, but also because right now in genomic technology and computational algorithm development, cancer field really benefits from the cutting edge of these new development. All the newest techniques, all the newest computational methods are being applied in cancer genomics, functional genomics, and precision medicine and cancer immunology. That's why we are gonna have a pretty heavy coverage. We're gonna teach you about a US consortium to generate tumor profiles of over 10,000 tumors, how to look at subtypes of one cancer type, how to look at survival analysis. For precision medicine, we want to kind of show you how to analyze drug screens and genetic screens such as CRISPR screen data, and also look at tumor samples to understand how tumor evade the human immune system, and how immunotherapy is really helping to cure cancer. And so that's the last module. So the course website is available from this link, but this is only available if you are within Harvard. We will have the earlier years of lecture videos available. I apologize, actually slides are no longer available because I think Canvas changed the link location and they removed the files. And so I will try to make the slides available before the lecture and post them onto the Canvas website. There have been video tutorials created previously from earlier years of students, and so we will have a link there. You can also look at the links to the previous labs. We will also record this year's lectures and labs. For discussions, we are not going to use Canvas because we realize there are students who might be interested in taking this course from anywhere else in this world. They may not have access to to Canvas, but they might still want to join some discussions. So we're going to have the course discussion on Slack from this link. I'll also put this link on the course Canvas website. So if you want to take the course remotely, or if you have a time conflict with this course, it's okay. We're going to record all the lectures and put them on the Canvas website. And this year, I also will create a Git book. And this link is already available. We have a prototype textbook, which is currently linked to previous years of lectures, and we will gradually be updating them. And because this year, the whole course is on Zoom, instead of giving many bad lectures throughout the semester, I'm only going to give maybe a third or half of the lectures to live to be updated. Some of these lectures can also be given by my colleagues, not from Harvard, from elsewhere in the US, who are better expert in this area than me. And so you can really get the best from all the faculty in this field. But we will keep some of the old lectures there. I am aiming to improve this course gradually over the years, so we will only update maybe a third or half of the lectures every year. And so on the days that we will not have live lecture, I will post the old lecture videos online ahead of time and send an email announcement. So students will know that that day we will not have a Zoom lecture, and you can just watch the previous years of videos. And so just watch for your Canvas email announcement. This course will have 6 homework. You will submit the homework on Canvas. This is the Harvard website. And for each homework you are going to submit, make sure you include a R markdown and also the HTML output. Write down your name and the course that you are taking, whether it's STAT115, 215, or Bio 282, and then which homework it is. So out of the 6 homework, homework 1 will be 9 points. This is the same for undergraduate and graduate students. I would expect this homework to take roughly 10 hours on average. If this is taking you way more time than this, you might consider whether to get better prepared for taking it in future years. Homework 2 to 6 are going to be more material. We expect it to have about twice as much material, but it will count 15 points for the undergraduate course and 20 points toward the graduate course. We will distribute just one set of problem set for each homework, but on questions that are labeled for graduate student only, that means that if you are only taking STAT115, you don't have to do those. But students who are taking the graduate level of the course, you have to do all the questions in that problem set. And that's why graduate course have more points. At the end of the course, we will curve the undergraduate students and the graduate students, the students taking the 200 level courses separately, because your homework are slightly different. But we'll make sure that the undergraduates will kind of cover a subset of the graduate material. That said, if you are taking STAT115 and you want to really try it out to answer all the questions in the homework, you're welcome to do that, and we can give you some bonus points there as well. After each homework, there is going to be an online survey directly on Canvas, and we hope all the students give us feedback on how well you feel the lecture is running, the labs are running, and how many hours did you spend on homework. There are some questions that are too hard or too vague, or took you too much time to do. We would really love to hear from you on that, which will help us improve this course for future years. And so as a small incentive, we give students 0.2 towards your final course credit. And so these are not added up to 100, but we will scale it to 100 points at the end. The homework survey is 0.2 points each. At the very end of the year, we're going to have an optional credit, about 5 points, to ask students to help us design a new question for next year's homework. This way, we will have some new questions for students. It's not an easy thing to do. I'm hoping that later on you will see from this doing the homework, we try to use some exemplary data to show you how to use different algorithms to touch on all the topics we covered in the lecture, so you can see how the different material fits together into each other. In order to create this homework, it's actually a lot of effort. And so unfortunately, we cannot update all the questions every year. It just takes too much effort, but we're hoping that through this, you can see how it's done, which means if you want to do the homework, we encourage students to discuss together, but you have to turn in your individual homework. You cannot turn in team homework. You need to submit your individual one, but you can discuss these questions on the Slack. There are a lot of trial and errors involved, and we really hope students can help each other. So as we mentioned, homework 1 is released today, and it's going to be due in 2 weeks' time. All the remaining homeworks, usually it's spaced at 2 weeks apart. This year, one thing is different. Normally we have a whole week in March of spring break, but this year there are 5 different wellness days. And so in those days, there are no lectures. And so the first homework is due on Sunday midnight. But I think as we see more and more wellness days, some of the homework might be due on Tuesday or Wednesday or Thursday. But we will give students 6 late days throughout the semester. You can choose to use 1 day on each homework, or you can choose to use 2 late days on the last 3 homework or whatever. But you have 6 late days throughout. We ask that you don't take more than 3 days on one homework, because we have to leave time for the TA to grade the homework. By the way, if you take additional late days, after you use the 6 days, each additional late days will incur 10% daily penalty. And we really don't encourage students to wait too long. After you use, say, 3 days on a homework, 3 late days on a homework, it's time to move on. Submit whatever you have, because the next homework is coming up very quickly. And so it's time to kind of work on the new questions. For labs, we have regular labs where the TA will really show you, like command line, you can see the screen, or, show you how to run something, how to do some specific type of coding on Tuesday at 6 p.m. and also Thursday at 8 p.m. These will also be recorded. And if the Tuesday recording is done very well, the TA will post the tutorial video on the website as well. We will also make those available on the Gitbook. If the recording is not done very well, the TA might record again on Thursday. But if it's recorded well, they will send an announcement. So Thursday will just be student asking questions. And on the weekends, we will have office hours. Those will not be kind of lecture style labs. You can watch the video recording. That's mostly kind of like an office hour to help students with homework questions. So the labs, well, as we mentioned, the lecture style lab will be recorded as well. And the Zoom information is also on Canvas, but the recorded videos will be put on YouTube and be available on both Canvas and the Gitbook. One important thing on the Canvas is that we need all registered students to sign up for a Canon account. This is the big Harvard cluster. You will need a Canon account to do the homeworks. So please sign up today so we can create the Canon account for you as soon as possible. And in the first lab, the TA will teach you how to log in, how to move around between directories, how to submit jobs, especially if some of the jobs are time consuming, how to run them in the queue. And so lab will start this week, starting Tuesday tonight. And so make sure that you get your Canon account so that you can go to the lab and start learning right away. And so the labs are going to be super, super useful for homework. They are going to teach you how to run a lot of the software, how to do a lot of the coding, and the TAs will help you answer questions. We will try to, we understand that this lecture style lab are only available in the evenings because during the day, the TAs also have to take their respective classes. But on the weekends, when we have office hours, we will try to have office hours in the morning as well, in case you are in a different time zone. Okay, I think that's all the information for the course, and I hope you will decide to take the course and enjoy learning. It's going to be a lot of work, but I hope you will find it really rewarding. There are, I hope that you will come back to this course later when your research topic is on this area. You watch the video, look at the homework, and really learn more material from the same course and hopefully understand even more. Okay, so good luck.